Dear participants, please wait for some time. We have some issue in network. As you understand the concept now. Good morning, sir. Sir, are you free? Can you join now? Ganesha sir is having some network issue. Participants, please be live. Sir is joining for next. Next sir is joining. Belavadi sir is joining. Yeah, sir is joining now. As Ganesha sir has internet issues, short while we'll be having next speaker with us. Please wait for 2 minutes. Good morning, sir. Sir, you can start sharing your screen. Sir, you can start. Morning, sir.
Good morning. Yeah. Shall I start now? Hello?
Yes, sir. You can start, sir. Please.
Yeah, in today's lecture, I'll be speaking on bees, the major pollinators of crops and wild flora. I will also be touching a little bit on uh, the floral phenology, why it is important, and I will explain that with the help of an example. So you must remember that in my last lecture, I was speaking about the evolution of plant pollinator relationships, pollination syndromes, types of pollination, and agents of pollination. And we all know that 87.5% of all flowering plants depend on pollinators. Now, in today's lecture, I will be speaking, as I just informed, uh, about the bees, a brief introduction to bees, how many species of bees are there in the world and how many we have in India, and what are the different families of bees. I'll be just touching upon these things. And then the link between plant phenology and pollination, why we need to study the plant phenology, especially the flowering phenology of plants with respect to pollination. And the pollination biology of cardamom I'll use as an example to explain about the methodologies and why it is necessary to conserve bees. To give you one more impression on the importance of pollinators, there was a very interesting paper published in 2011 by Parthiba Basu and his group from Calcutta University, in which they actually covered the entire, say, the cultivated plants. They divided into three groups as pollinator dependent crops, pollinator independent crops, and crops with which are highly dependent on pollinators. And they interestingly found that over the years from 1960s to 2010, they found that there was a steady increase in productivity of the crops which are pollinator independent. That means because of the efforts by the breeders, they could actually develop varieties which are high yielding. So all those cereals that we consume almost every day, you see that there is a steady increase in the productivity and also the production of these crops. But the same was not true with the crops which are dependent on pollinators, or the crops which are highly dependent on pollinators. There was a plateau in the yield from in 1990s onwards or 1980s onwards, because probably they argue that we did not consider the importance of pollinators for these crops, and that's the reason why we have not been able to increase the yields of these crops. So that is the main problem. So coming back to the question of whether we are ignoring pollinators, there was a very interesting book published in 1996 by Bookman and Noben, The Forgotten Pollinators, which in fact opened the eyes of many scientists and also the policymakers about the importance of pollinators. And I think all of you should read this book. It is a very, very, very interesting book. And again, in the early 2000s, there was a special issue in Science on the pollinators. So these are the important publications which actually started or gave a lot of, say, force for the study on pollinators. Now, when we hear the word pollinator, we think of only bees. And in the last lecture, I think I also told about what are all the different pollinators that are there other than bees. But we consider bees as major pollinators because we see more often plants being visited by bees. And when we hear the word bee or hear about bees, we think of only honeybees. That is another myth that we have. In fact, there are more number of species of bees, more number of species of honeybees in the world than the number of honeybees. We have only about a dozen species of honeybees in the world, and in India we have only 5 species of honeybees. But we have a lot more number of bees which are not honeybees, which are also important as pollinators. Now, the question is how many bees we have. In the world, we have something like 20,507 species of bees in the world as on today. Okay, and the estimate is there must be more than 25,000 species because almost every year or every month you find publications that are coming out with new species of bees being discovered. In India, we have 723 species, which amounts to about 3.5% of the world bee fauna. And probably we have many more species, and there is a greater task for the bee taxonomists to look for new species of bees and their importance in India. So the bees come under the superfamily Apoidea. There are two groups in the superfamily Apoidea, Specieformis and Epiformis. Specieformis includes all spicoid wasps, which are carnivores. That means they feed on other insects. They also build nests like the bees. They feed on, they predate on other insects or spiders and feed their young ones with this carnivorous diet. And these are supposed to have evolved from species wasp-like ancestors. Probably during the course of evolution, they changed in fact, the species wasps also, they visit flowers for their own survival, the adult, their own survival. They take nectar, they feed on nectar. And probably bees also initially, they were evolved because they were visiting flowers. And they started feeding the young ones also with the rewards that they started getting from the flowers. So that's how probably the bees have evolved, and there is a close relationship between these two groups. Now, all bees, I said, they belong to the group of Epiformis, and there are 7 families of bees: Andrenidae, Apidae, Collectidae, Halictidae, Megachilidae, Melitidae, and Stenotritidae. And of these 7 families, we have in India 6 families. Stenotritidae does not occur in India. And Melitidae, it is represented by only 4 species in India, whereas the maximum number of species we have are Megachilidae, Apidae, and Halictidae. These are the ones with highest number of species that we have in India. And all these are excellent pollinators of several crops that we have, or several crops and wild species of plants we have in India. And most interestingly, of these 723 species of bees we have, 44.5% are endemic. That means they're found only in India. And supposing if we have some important species of bees among these 44 or 45% of bees, and if we do not conserve them properly, and if we lose them due to some reason, we can't even import from some other country. So it is important to know what bees we have and what is the role of these bees for our own crops and our own wild plants, and why we have to conserve, or how we have to conserve these bees. Now, this is based on Asher and Pickering 2021, and we also have a paper similar to that. So some of the bees, solitary bees, I told that there are nearly 723 species of bees in India which are solitary, and only 5 species of bees are honeybees, which are social. Okay, so Thyreus ceratina, and there are species like Megachile and Xylocopa. There are several species in India. And we also have one very interesting group of genus, which is Amegilla, which is the excellent pollinator of many crops, including solanaceous crops that we cultivate. Now, coming to the phenology, why phenology is important. To know about what is phenology, it is the timing of changes in plant and animal life stages. That means for a plant, as far as a plant is concerned, when a plant enters into the reproductive phase, when it starts flowering, this is very important from the point of view of success of pollination if it is dependent on a pollinator. Okay, so the new foliage and flowering is a phenological stage. Emergence of insects with reference to insects, activity of pollinators. That means when the flowers are available, the pollinators should be active. So there should be some kind of relationship between these two. And then, of course, migration of birds. All these are considered as phenological events. Phenology actually refers to the responses of living organisms to seasonal and climatic changes. So over several decades, several thousands of years, they each species would have got adjusted to certain changes, certain seasonal happenings or environmental conditions, and would have started acting accordingly. So that is the reason why you have in several species of plants, the flowering occurs in a certain season. The flowering may not occur throughout the year. It occurs only in certain months of the year. So it is because it is adjusted to flower in such and such a condition. And each species or sets of related species of plants flower only in a given season. But there are a few exceptions. One of the exceptions is, of course, the fig, which actually flowers throughout the year. And there also there is a different kind of a system where in a population, all fig trees will not be flowering at the same time. They will be flowering continuously. A part of the population will be flowering in 1 month, and in the next month there will be another few individuals will be flowering. So like that. So there are a few exceptions for this seasonal behavior. Now, what information is needed when we are studying phenology? Uh, from the point of view of pollination or the success of pollination, we need to know when the plant starts flowering or when the flowering initiation starts and when the flower opens. Once you find the flowers on the plant or buds on the plant, so when it actually opens, whether it is the anthesis, whether it is in the morning hours or in the evening hours or in the afternoon or maybe after about 2 or 3 hours after the sunrise. So all these kinds of things are important from the point of view of the pollination. And the time of anther dehiscence, when exactly the anther dehiscence or the pollen dehiscence occurs. So there are several adaptations for this. There are plants where the pollen dehiscence or the anther dehiscence occurs before the flower opening, where it is referred to as cleistogamous. So the pollen is ready even before the flower opens, and even the pollination would have occurred even when the flower is closed. So that is a self-pollination adaptation. And in some species of plants, the anther dehiscence occurs after the stigma becomes non-receptive in the same flower. Okay, so that there is no chance of self-pollination. Or first the anther dehiscence occurs and the stigma will not be receptive in the same flower. So all these are adaptations to prevent self-pollination. And then pollen viability is another important thing. So once the anther dehiscence occurs, pollen comes out of the anthers, how long the pollen grains remain viable. That means what is the time span that is available for a pollinator to transfer this pollen or the pollen to reach the right stigma. So this is also important. And then of course, stigma receptivity. Stigma receptivity is when actually the stigma is receptive, and when the pollen is viable, when the pollen is transferred by the pollinator, the stigma should be receptive. And of course, nectar production, the time of nectar production, and the quantity and quality of nectar production. So these are the three important things which actually determine the pollinator activity also. So pollinator activity should coincide with the pollen viability, stigma receptivity, and also, of course, the nectar should be available. Otherwise, the pollinator will not be visiting the plants. So this leads to the pollination success. So I'll take an example to explain how the phenology is important to with reference to the pollination success. So how phenology and floral biology and pollinator species and its behavior are linked. I use the example of cardamom, okay, on which we have worked. I have some first-hand experience on this. That's the reason why I take this example. Cardamom is the queen of spices, Elateria cardamomum. So this is the flower of cardamom. It depends on bees for pollination and and it is a commercial spice crop cultivated in the Western Ghats of India. It in fact originated in Western Ghats of India. The place of origin is Western Ghats of India. Now, these are the regions where the cardamom is cultivated. All the ones which are marked yellow, this is the region where in the Western Ghats, in Kerala, Karnataka, and parts of Tamil Nadu, the cardamom is cultivated. Now, if you get into a cardamom field in the month of July or August, in June or July, you see the cardamom field like this, where the cardamom, the panicles are at the ground level, and you see that the flowers and the capsules at the ground level. So each plant of cardamom is referred to normally as a clump of pseudostems. There are several pseudostems, 20 or 25 pseudostems, and from each pseudostem there will be one or two panicles that come out, and on the panicles you have the flowers, and the flowers, if they are visited continuously by the bees, they will set into capsules. So that is this is how a cardamom field looks like in the actual season. And cardamom, as I said, is a zingiberaceous plant. It belongs to the ginger family and is cultivated for its most valued capsules under the shade of trees. The shade trees are original forest tree species and are specifically planted for the shade. Now, the shade trees are very important. In addition to providing shade for the cardamom plants to grow, shade trees also have flowers, and they should be flowering in the season when the cardamom is not flowering, so that they can keep the pollinators intact in the field. So for conserving pollinators, conserving shade trees also is important. So this is a cardamom clump with capsules and with the panicles. And in India, cardamom is cultivated in 2019-20. If you look at the data, it was cultivated in an area of 69,000 or 70,000 hectares in the southern states of India, mainly Kerala, Karnataka, and Tamil Nadu. Nearly 60% of the area is in Kerala, about 30% in Karnataka, about 10% in Tamil Nadu. Now, it is very important because in the last year itself, the total production was 11,230 tons, and of which nearly 2,000 tons were exported, fetching nearly 426 crores in foreign exchange. So it is a very important income earning crop for the country. Now, it grows in clumps. As I said, cardamom plant is referred to as a clump of cardamom. It grows in clumps of 25 to 30 pseudostems. Each one is a pseudostem. And at the base of the pseudostem, it bears 2 or 3 panicles. And the panicles bear the flowers, and the flowers turn into capsules. So this is the how a cardamom plant looks like. If you have not seen the cardamom plant. Now, there are 3 types of cardamom. The first one is referred to as a Malabar type. Malabar type has the panicles which lay prostrate on the ground. They are at the ground level. The Malabar type is the panicles are prostrate on the ground. And the next one is Mysore type, which has erect panicles. The panicles are erect. Now, interestingly, the Malabar type is cultivated in Karnataka, and the Mysore type is more common in Kerala. So this is I really do not know how it happened. There is an intermediate type also, the Valuka type. The Valuka type is semi-erect. It has semi-erect panicles. Probably it is a natural cross between the Malabar and the Mysore types, so nobody knows about that. And in fact, some of the high-yielding varieties of cardamom are from the Valuka type today. Now, when you start working on the pollination biology of any crop, some of the questions that one should ask is about the floral biology of cardamom. So what is the floral biology of cardamom? When does it flower? What is the season of flowering? What are the months in which the flowering occurs? Then what is the flowering duration? How long? Supposing if it starts in the month of April or May, how long it continues? How many months you have the cardamom flowers? Because that is important for the activity of bees also. And when is the anthesis? When the flower opens in the day, and when is the pollen dehiscence? Then what is the pollen viability? How long the pollen is viable? When is the stigma receptive or the peak receptivity of the stigma? And how long the flower remains? Once the flower opens, how long it remains, whether it remains for a single day or whether it remains for more than a day. So this is another important thing. And of course, the flower structure. Flower structure is important from the point of view of the uh, pollinator, because it has to recognize the flower, it has to land on the flower, and it should know where the nectar is available, and then in the meantime, it should also transfer pollen. And then you also should know what are the insect visitors, which are all the insects that visit. Now and their foraging behavior. What is the behavior of these insects which are visiting? Whether there is whether they visit in a single visit, whether they are visiting several flowers, or they are visiting several species of plants or the same species of plant. So all these are important. Now, the first and the foremost thing is to find out how many of these questions you already have answers in the literature. Because it's not a new plant. People must have worked on this earlier also, and they must have looked at some of these things. And we have to look at the literature to see how many answers that we have for how many questions. So we found that for some of these questions there are answers. Like when does it flower? There was a uh, there was some information that what is the flower duration. There was information in the papers that were published. And when is the anthesis and pollen dehiscence? There was information. Pollen availability, there was information. Longevity of the flower, it was already known that the cardamom flowers last for a single day, and then the flower structure, there was information. But the information that we had for some of these things, there were still some question marks. Like the season and months, there was some variations in the papers also and in our own observations. And then when is the anthesis? It was simply given as in the morning hours. And then pollen viability, there was again some very interesting say error in that, which actually made us to work on that. And then about insect visitors and their foraging behavior, there was absolutely no information. And stigma receptivity, there was no information. So there was a lot of things that we should be able to work on. So we started. So the available information was this: the flowering May to September, October, and the peak in July, August. Anthesis morning hours, pollen dehiscence morning. Then pollen viability is only 2 hours that was given, and then the stigma receptivity, there was no information. The flower lasts for a single day, and the important question here was the pollen viability was 2 hours. If the pollen viability is only 2 hours, and if the pollen dehiscence is in the morning, that means if the pollen dehiscence, if the anther dehiscence occurs, let us say at 7 o'clock in the morning, it should get transferred before 9 a.m. And during that time, the pollinator, the bees should be active, the bees should be visiting. So this was a serious problem that we had, because we did not see much activity before 10 o'clock, before 9 o'clock or so. So we wanted to look into the details about this. So we looked more carefully about the floral biology. The floral biology of cardamom, the cardamom flower has 23 millimeters long corolla tube. The corolla tube is 23 millimeters long, which actually holds nectar. The corolla tube, when it is full, it holds about 4 microliters of nectar. And the flower also offers a lot of pollen to the pollinators. And there is an enlarged flabellum which acts as the landing platform for the bees. And on the flabellum, you also see that there are nectar guides. So the bee has no difficulty in landing on this and then knowing where the nectar is available. So it pushes its proboscis inside this corolla tube to take nectar. And in the meanwhile, the stigma is here, and the anthers are below, the pollen grains are below. So there is no question of self-pollination here. But when a bee comes after visiting another flower and then comes and then lands here, it will be carrying pollen grains on its head, and that will come in contact with the stigma, and then effective pollination occurs, or the deposition of pollen grains occur. And when it is going back, it takes pollen again from the anthers, and then it goes to another flower. So this is how the pollination occurs in cardamom. Then, so that is how the bee lands and then takes nectar. Now, if you look at the distribution of flowering in cardamom, you see that the flowering actually starts in the month of April itself. In the April third week itself, we started observing the flowering. And then the peak is in July, August. That is the time when you have the maximum flowers that are produced, and it continues till November. So from April second or third week, the flowering starts, and then it continues till November. but the peak flowering is in July, August, and most of our initial observations on the cardamom pollination were restricted to the peak flowering. I'll tell you later a little bit about that. So the flower density, if you look at, you see that in April and May and June, the density is low, okay? And this is the total number of flowers in a month. So every day when you go to the cardamom field, you find new flowers. And when you record all the flowers per clump, which is produced per day, this will be the number that you get. And in August, September, you find the maximum number of, in July, August, you get the maximum number of flowers per clump. Okay, that is a peak flowering. Then if you look at the pollen viability, the data that we had, the earlier literature said that the pollen is viable only for 2 hours, but we found that the pollen is viable throughout the day, but the peak viability is around 12 noon. So this is one information that we got. So the methodology, the difference in the methodology between the previous workers and us was that the previous workers, what they had done was they had collected the flowers soon after anther dehiscence or pollen dehiscence in the morning, early in the morning, around 7:30 or 8 o'clock in the morning. And then they had brought it to the lab and then kept the flowers in the refrigerator. And then at every 2 hours, they had taken the pollen grains and then looked at the germination percentage. And they have found that the pollen germination occurs only for 2 hours. Probably the refrigeration might have affected the germination or something like that. And probably that is the reason why they got that kind of a data. But what we did differently was every 2 hours, we had covered the flowers in the field so that no bee visits those fields. Several hundred flowers we had covered like that. And then every 2 hours, we used to get fresh flowers, all of these closed flowers, and then we used to take out the pollen and then put it for germination. So we found that the pollen is viable almost throughout the day, and maximum germination occurs or maximum viability we found around 12 noon. Then coming to stigma receptivity, we actually did some hand pollination studies at different hours of the day, and then we find that the stigma also remains receptive almost throughout the day, and the maximum receptivity is between 11 and 1 p.m. So this is the maximum receptivity. So the pollen viability, the maximum pollen viability is around 12 noon, and the stigma receptivity, maximum stigma receptivity is between 11 and 1 p.m. So that is the second thing. Then nectar accumulation rate. If you look at the nectar accumulation rate, maximum nectar accumulation occurs around 11 a.m. Nectar is available throughout the day. The nectar production is continuous in the flowers, and the rate of accumulation starts increasing from 9 a.m. and then it is maximum around 11 a.m. and then again it comes down because of continuous harvesting of the nectar by the bees. Now, under foraging activity, if you look at the activity of bees in different times of the day, you see peak activity between 11 and 1 p.m. Now you see that there is a lot of, correlation between these 4 things. One is pollen viability. Pollen viability, the peak is pollen viability, stigma receptivity, nectar secretion, and bee activity. You see that all these, the peak is between 11 and 1 p.m. So there is every reason to believe that bees are efficient pollinators by just looking at this data. And of course, we did a lot of other experiments with closed flowers and then allowing only bees to visit, and then what is the percent set and all that. So this is how the phenology of a plant can be used for studying the pollination of any plant. So when we rewrote the floral biology of cardamom, the flowering, the earlier information that was there was May to September, October, but we found it from April to November, but the peak is in July, August. Anthesis is between 4:30 and 6:30 a.m. Pollen dehiscence, it is around 7:30 a.m. And pollen viability, it is viable throughout the day. Stigma receptivity peak around 1 p.m. and flower lasts for a single day, which is the information that we already had. Now you look at the relation between the number of flowers per clump with the number of foragers per clump. As I said, clump is one plant. A clump is a clump of pseudostems, which is considered as one plant. Now, with the number of with the increase in the number of flowers, the number of bees active per clump also is more. That means when the number of flowers is less than 5, there is no activity of bees, and when the number of flowers increases, the number of bees active on the clump also increases. So there is a perfect correlation with that. And a single forager visits more than one flower. Okay, it visits more than one flower because with a single flower, it will not be able to completely fill its stomach. So the stomach capacity of a bee, a honeybee, an Indian honeybee, is about 40 microliters. Now, even if it is getting the full 4 microliters of nectar, it has to visit about 10 flowers, but it is not able to take all the 4 microliters in any flower. Though initially in the first trip it may get more nectar, so it visits about 40 flowers, and then in the second visit, these are all done with marked bees, individually marked bees, which we could actually recognize in the field that it has come for the second trip or the third trip or the fourth trip and all that. So a bee makes about, on an average, about 5 to 7 trips, and after about third trip, the number of bees that the number of flowers the bee has to visit per trip increases to about 100 to 120 flowers. So it has to visit so many flowers because in each trip, with a competition for the nectar, more and more bees will be visiting, and in each flower, a bee will be getting only about something like zero point 0.3 microliters of nectar. So to fill its stomach, it has to visit at least about 100 or 120 flowers. So this is one very interesting thing, because unless a bee is made to visit more number of flowers, and more number of times the same flower, the pollination will not be effective. Because in a cardamom flower, the number of ovules is about 27. There are 3 locules, and in each locule there are 9 ovules, and 27 ovules are there, and each ovule should receive a pollen grain. So in a single visit, it will not be able, a bee will not be able to deposit so many pollen grains, and even if it deposits about 30 or 40 pollen grains, it will not be effective because there will be competition between the pollen to reach the ovule, and all the pollen grains will not be successful in reaching the ovule or fertilizing the ovule. So that is the reason why the flower remains attractive almost throughout the day, and it remains attractive to the bees, and the bees will be continuously visiting the same flowers, okay, repeatedly. And so the number of visits to a single flower will be definitely more than 20 to 30 per flower. Okay, so that is how the pollination effectively takes place. Now, the total number of flowers visited by a single bee per day is in the beginning. These are individual bees which are marked by, we had given the numbers for them, and so it varies from about 250 to some visit about 700 flowers with so many trips it makes. So some 3 or 4 trips it makes, and then it may, or even 7 trips it might have made, and it has visited about 700 flowers. So it is greatly variable, but on an average, a single bee visits at least about 100 flowers. And a single visit, the number of visits to a single flower in different times of the day. So again, between 11 and 1, you find, or 11 and 2, you find the maximum number of visits to a single flower. Okay, the single flower is getting at least about 20 or 25 visits around 12 or 1. So this is between, we see that between 11 and 2 is the time when the maximum pollination occurs. And the relation between the number of bee visits per flower with the number of pollen grains deposited on the stigma, there is a relation. With the increase in the number of visits to the single flower, the number of pollen grains deposited per flower also increases, and this is also reflected with the number of fruits set with the increase in the number of visits. But on an average, if you look at the fruit set in cardamom under natural conditions, it is only about 30% or even less. So if 100 flowers are there, only about 30 of them will be actually setting fruit, setting capsules. The fruits of cardamom is referred to as capsules. So it's setting capsules. So that means to say that there is some kind of a deficit with the number of pollinators available or number of bees available. So the number of bees may be less. We may have to add the number of colonies per unit area. The natural colonies may not be sufficient uh, for actually pollinating all the flowers that are available or maximum number of flowers available. And in fact, there is a different thing which I am not presenting today. By introducing colonies, we can actually increase the capsule set. Another interesting thing is, for decades, it was believed that cardamom is solely pollinated by honeybees. There are many papers, including our own team. And the honeybees were Apis cerana and Apis dorsata, the Indian honey hive bee, and also the giant bee. These are the two species which are most commonly found when the cardamom flowers are there, or when the cardamom when we see the cardamom flowers. And we also found that with the number of flowers available per clump, when the number of flowers is less than 5, honeybees do not visit cardamom, because it is not economical for a bee to visit flowers of cardamom when in each plant there are only about less than 3 or 4 flowers. Because if it has to fill its crop, it has to visit at least about 100 flowers, I said. So to find 100 flowers, it has to visit some 20 or 30 plants in search of the flowers. And that actually, it is actually spending a lot of energy in searching only, and spending energy whatever it is getting from the flowers. So that is the reason why they neglect cardamom when the density is low. When less than 5 Apis cerana does not appear, when the number of flowers per clump is less than 5, and Apis dorsata does not appear when the number of flowers is less than 15. Only at the peak flowering, dorsata comes, and when the near to peak, the cerana will be active. So in July, August, or in late June, July and August, you find only Apis cerana and dorsata, the giant bee and the hive bee, active on cardamom. But what happens to all those flowers which are produced, which are borne by the plants before this, in the early season or in the late season? So this was a question that we wanted to answer. So we wanted to look at the relation between the flower density and bee visitation, flower density in different months of the flowering season, and the species of flower. Are there any other species of insects which are visiting the flowers in other months? At the diversity of these species, diversity and abundance of these species. And we also wanted to look at the tongue lengths of these species. There is a reason for this. I'll come to that. Now, the flower density in different months, you see that I have already told this. In April, May, June, the density is very low. In July, August, it is very high. This is the number of flowers available per day per clump. Up to about 25 to 30 flowers you'll find in August. In July, you'll find between 20 and 25 flowers. And again, in September, October, it becomes low. So this is the period where we call it as the peak flowering period. Now, the flower visitors that we came across were several species in different months, in the early season and in the late season. There were several species of Amegilla species, Serratina species, and including Tetragonula iridipenis, and also, of course, Apis cerana and Apis dorsata. So all through we were concentrating on only these two species, but these were also visiting in the seasons in the time when the flower density was low. You find several other species of bees. All these are solitary bees. Now, if you look at the bee diversity in different months, you see that in the month of May, when the flower density is low, you find one species of Amegilla species 1. There are 4 species of Amegilla we have recorded. Amegilla species 1, maximum numbers you find in the month of May. And in the month of June, you 4 species of Amegilla, still you do not find cerana there. And in the month of July, you find Apis cerana and very few of Amegilla species. And in August, you find cerana and dorsata. Dorsata takes over. And in September, you see again Amegilla species and cerana being active continuously. So this you see that the flower density, bee diversity, and fruit set are also very interesting. The thing is, number of, if you look at the number of flowers setting fruits, the percent flowers setting fruits, you see that in May and June and in the late August and September, you find more number of flowers setting fruits. Percent setting is higher compared to July, August, let us say July, August, when the honeybees are more active. So this is the time when Amazona species are active, and this is the time, July, August, it is the Apis species that are active. So the number of flowers are more here, the number of flowers are less here. Apis does not visit here, Apis does not visit here, and it is only the Amazona species that are visiting here, and they are responsible for maximum fruit set here. But here the number of flowers are more, but still we get higher yield or higher fruit set. The person, though the fruit set percentage is low. So you see that the Amazona species appear to be more efficient because the percent fruit set is very high. So is there a reason for that? We wanted to look at that. So when you look at the flower of cardamom, you see that I think you can see that there is a line here. This is the level of nectar. Okay, so this is the corolla tube. This whole thing is the corolla tube, and when it is full, it is supposed to have 4 microliters of nectar. And if you go to the cardamom field during the peak flowering season and then pick any flower, you see that the flower is about 50% with about 50% nectar. Okay, this is about 12 millimeters or 11.5 to 12 millimeters nectar. So almost all flowers will have 11.5 to 12 millimeters nectar, or about half of the corolla tube still with nectar that remains unharvested during the peak flowering period. Now, the 50% of the nectar is unharvested in every flower, and you know that the nectar production is costly. So nectar involves a lot of sugar, and it is very costly for the plant to produce so much of nectar. And if the nectar is not being taken by the plant, by the pollinators, that means there is no point in producing this nectar. It's a wastage for the plant. Though it may be reabsorbed later on, but still there is a lot of waste that is going on for the plant. Now, at any given time of the day, you see that the nectar level, it remains around 11.5 millimeters. Okay, the nectar level is, so this much of nectar is again going waste. So only above this, whatever the nectar that is secreted above this is only harvested, and the remaining nectar, the bees are unable to harvest. Bees means the honeybees are unable to harvest. Now, why is that so? If you look at the tongue length of honeybees, both Apis cerana and Apis dorsata have significantly short tongue lengths compared to the corolla length. Okay, the tongue length of both cerana and dorsata is between 4.5 and 5.5 millimeters. The corolla length is 23 millimeters. So with 23 millimeters of corolla length, with full of nectar, the tongue with a short tongue length of 4.5 millimeters, the bee is still capable of harvesting, taking nectar up to 11.5 millimeters. And that is a question, that was an interesting question. And in fact, this was done by one of my students who actually discovered something very interesting in this. So we actually used artificial flowers, which we made using capillary tubes. 23 millimeters long capillary tubes we took, and the bottom of the capillary tubes was closed by beeswax, and then this was filled with sugar solution. So before that, we trained the bees to take sugar solution from the capillary tubes, and then we did a lot of training experiments, and then did these experiments. So when the capillary tube is completely filled and a bee is allowed to take nectar from this, it actually took, this is 23 millimeters long, but it actually took nectar only about its tongue length, 4.5 millimeters. It did not go beyond this. Then my student came up with a very interesting observation in which he said that the style of the flower, it goes through the capillary tube. Okay, it goes through the capillary tube, and then it goes through the anther lobe, and then the stigma comes out above the anther lobe. So that is how the structure of the flower is. So this style that is inside this corolla tube, it might be helping the bees in some way. So how to test this? So for this, what we did was we used artificial styles. Okay, of thin wire and also some ixora flower, the styles of ixora flowers. And then we introduced that inside this and then saw how the bee takes, to what level it takes. So it is interesting that this is the experiment that we did. So this is the style that is there um, inside the corolla tube, and the bee takes the nectar. It does not even insert its say proboscis completely, and it started taking the nectar right from the top, and then it could take up to about 11.5 millimeters. So we found that this portion, it might be acting like an open-ended capillary tube, and probably there is through by capillary action it is going up, and there is a limit for that action, so it stops after some time. So this was an interesting paper we published more than 25 years ago. So look at the correspondence, and so the next thing is to look at the tongue length of Amygdala species. So the tongue lengths of Apis species do not correspond to the corolla length of Cardamom, but the tongue length of Amygdala species is very long. Amygdala has a tongue length of about 15 millimeters long, and it can take the entire nectar from the flowers. Okay, it is capable of harvesting the entire nectar. So one of the hypotheses that they have longer tongue lengths and are capable of exhausting nectar. So one of the hypotheses that we proposed was that Amygdala probably was the original pollinator of cardamom, because cardamom also originated in the Western Ghats, and there are several species of Amygdala in the Western Ghats. And the tongue length and corolla length corresponds, and their correspondence is there, and they are more efficient pollinators, and then the setting also is very high. And the honeybees might have replaced Amygdala in cultivated habitats. In the cultivated habitats, you have more number of flowers. The Amygdala being a solitary bee, the number of bees available is very less. So they will not be able to visit all the flowers that are available. And honeybees being opportunistic flower visitors or nectar harvesters, they take advantage of that, and then they actually compete with Amygdala, and then the Amygdala move out. So the honeybees actually will be more in the peak flowering seasons, and that's how we get higher uh, activity of honeybees. Now the next question is, what are the implications of this? In the cultivated systems, the flowers are in large numbers. There are low populations of Amygdala which are being solitary, and large populations of Apis because they are social, and 50% of the nectar remain unharvested in every flower when the Apis visit, or in when the maximum number of flowers are actually visited by the Apis, because the peak flowering season, it is only the Apis which are visiting, the bees which are visiting. So there is a cost for nectar production, and is there a way in which the corolla length can be reduced? So is there an implication for shorter corolla? So is there a short corolla cardamom? So we wanted to look at that, the short corolla cardamom. The short corolla type um, should be spending less energy for producing nectar, so less nectar production. So less energy spent on nectar production, saved energy may be diverted for reproduction and produce more flowers. And so if you have a variety or an entry in the germplasm collection with shorter type, that might give an say chance for looking at a high yielding variety. So this is what we thought of. So the corolla lengths we measured for 173 entries in the germplasm. We had the germplasm in a cardamom research station. We in fact went to 3 cardamom research stations, and then we took both in Karnataka and also in Kerala. And we got the corolla lengths, we measured the corolla lengths of 173 entries in the germplasm. And interestingly, we found when we plotted the distribution of the corolla lengths across the germplasm and also the released varieties, we find that very few had less than 18 millimeters, but then 18 to 20 millimeters and 20 to 22 millimeters, whereas many of them had 24 to 26 or beyond that. Okay, so we hypothesized that the high yielding varieties should fall in this group, and in fact the variety that we were working on all through the Modigere 1 it has 23 millimeters long, and it is about this within this. And so we wanted to look at what is the what is the corolla length of the high yielding varieties. So there are two varieties, Mudigere 1 and Mudigere 2, which have the corolla length of 23 and 19.2. Then this is the variety from Sakleshpur, Cardamom resuscitation 18.75 millimeters. So all these have earned the highest yielding variety, which is very popular in both Karnataka and Kerala, is Nelani. It has a corolla length of 17.6 millimeters. So this is the highest yielding. So in fact, this is a very interesting information that we have got, and we have passed it on to the breeders so that they can look at the high yielding varieties. This can be used as a character for recognizing a high yielding variety. So honeybees and non-apious bees are both important for fruit set in cardamom. Amygdala is possibly the original pollinator and is active during low flower density situations, responsible for nearly 20% of the total fruit set. So it is important that we have to conserve Amygdala also, because whatever is happening, whatever the flowers that are there in the earlier or in the late season, they also should set, and we will be losing nearly 20% of the production if we ignore Amygdala. So we have to conserve their nesting sites also. They are all soil nesting species. And high yielding cardamom varieties exhibit shorter corolla length. Corolla length may be used by cardamom breeders as a key attribute while selecting higher yielding varieties. I think that's it. Thank you very much. Yes, sir. If there are any questions, I'm ready to answer. Hello? Are there any questions?
Sir, actually, I have one question.
Yes, sir.
Regarding cardamom.
Yeah.
What is the characteristic phytochemical content of the cardamom? Means it has its own typical aroma and its own flavor.
Yeah.
So what could be the component, I mean to say, active component giving that particular aroma and flavor?
Oh, I don't know whether I will be able to answer this. I should find out and then tell you. I think a physiologist will be able to answer this. In fact, it has an aroma. I forgot. I know the name of the chemical, but I don't think I can recollect now. Okay. Yeah, that is the reason why it is used as a spice, right? Because of its aroma.
So, is there any impact of these aroma on insects? I mean to say, because anyhow, olfactory clues are there attracting these insects.
Yeah.
So that aroma is because with the smell, generally we sense fruit or what it
flowers
is.
will not have the same kind of aroma. Probably it is more, maybe from the anthropocentric point of view, it is more we do not get any aroma from the flowers, but maybe the bees may get. But it is more visual, I think, because it is very from a distance you can actually recognize the flowers, because it is whitish with pink markings on the flowers. So that's how the bees come. There may be some chemical cues also. We don't know. There must be some. Yeah.
Participants, please, you can put in chat box, or if you want, you can unmute and you can ask a question.
Please.
I have made unmute. You can ask questions if you can. You can post in chat also if you have a question. So, meanwhile, I have one more doubt,
Yes,
sir. Actually,
yeah.
These bees are there.
Yeah.
They are so violent. I mean to say, when they bite, even human deaths have also reported.
Mm-hmm.
So, is these bees going to inject any venom into the, I mean to say, flower while sucking the nectar or something like that?
Bees use their sting only for defense. Okay, supposing if you disturb them when they are actually on the flowers, in fact we can easily collect the bee and then mark them and all that. We have done that. Never we have been stung. when they are foraging, they usually do not sting unless you try to catch with your hand and then try to harm it or something like that. It is only a defense action. And in the near the nests, again, they will be more cautious. If somebody tries to disturb, then they will attack. So that kind is there. Otherwise, they are very harmless. Yeah.
So, regarding social behavior,
Yeah.
when one bee is getting violent,
Yeah.
many of the bees will come for the attack. What could
Yeah,
be
they
kind of
have
signal
yeah, they have
generally
what
they
is
will
called
be?
they have what is called as a alarm pheromone. So
Yeah.
if a bee comes and then stings one person, it also releases a chemical, and that chemical attracts other bees to come and then attack the same person. So that is how it is. It is an alarm pheromone. And in fact, when a bee stings, it loses its sting. So that is another. It is suicidal for a bee to sting a higher animal. Okay, it loses its sting and then it dies. So very rarely they will be very careful, and unless it is very harmful to the colony, they will not use their sting.
Really, it is a
Yeah.
big lesson that how society might have
Yes.
developed. A
Yes.
single man in the
Yeah.
old Stone Age.
Yeah. True.
Srinivas, you need to ask some question. Dr. Srinivas. Yeah, it is a
Yeah.
good, it is a concept
Yeah.
how civilization has made,
Yes.
to
Yes.
say,
Yeah.
how, to say, a single man was a hunter once upon a time and
Exactly.
remained, he become a colonist and he become a societal. So this is
Yes.
a beautiful, wonderful, to say, concept that how bees can
Yeah.
construct a
Yeah.
big, to say, shelter for them and collect the things.
That is an entirely a different subject. How the evolution of social behavior developed. That is a separate subject. That is very interesting again. You know. You know that all bees are worker bees. Worker bees are sterile females, okay? And the male bees do not work. The drones do not work. There will be very, very few in the colony, and there will be a single queen which remains in the colony laying eggs. So it is the workers which come out and then collect the pollen, nectar, and all that, and then rear the young ones of their queen. Okay, so what they are rearing uh, is actually a group of sisters that they are trying to rear. So there is a very interesting study on how the genetic relatedness make them work for the colony.
Can we go for one more lecture, sir? Because sir is having, say, internet
Oh,
issues.
no, I'm not prepared. I thought it is tomorrow. And in fact, I wanted to speak on two more crops tomorrow. Okay. And day after tomorrow, I'll be speaking on fossils. I think you mentioned about fossils the other day.
Yeah,
So
yeah. One
how the work
participant
on
is asking.
fossils is done, and then how um, the um, the date of fossil is measured and then determined and all that. So those things I'll be speaking. Okay, sir. If there are any
Thank you,
more
sir.
questions,
Thank you so much for joining
thank you,
us.
thank you,
Please,
sir.
participants, wait for some time. I will start sharing one of the presentation for you. Thank you, sir. Thank you so
Okay,
much.
sir. Thank
Please
you.
stay.
Bye.
Please stay with us because our time is 12:30. So I will take one half an hour more. And the thing is, please participants, you use the same previous link. I don't know what was my mistake because my ignorance made today you feel some troublesome because I couldn't generate proper link of Zoom today. So different new link was generated. In afternoon session you will have again the same old link what you are using 73 number, so it will be no difficulty. So inconvenience is degraded for what happened actually.
Follow me, Nick Offer, as I take incredibly 8% of all insects live in jungles. Fewer more.
Hope you're able to see this YouTube video. Are you able to see, please? Somebody can reply. Hello?
Yes, sir. We can
You're
hear.
able
We
to
can
see,
see.
you're
Yeah,
able to
it's
see
visible.
one video
It's
of
vis-
BBC?
yes, it's visible.
Okay, thank you. Let me start. Let me start one concept. You will be seeing the video. So in this video, my talk, I'll talk on something. What is this video actually? Please, you see this video of 3 minutes. Then I will start.
80% of all insects live in jungles. Fewer more successful than the average. There could be 8 million individuals in a single
I was mute actually. So what I was trying to do is I was trying to show you the video. In that video, I was trying to show you the concept. What was the concept is there is an ant and there is a fungus. What is happening with ant and the fungus? Actually, I am going to describe that because we were talking sometime about ecosystem services. We were talking about ecosystem services. What do you mean by ecosystem services? That I will talk. But before that, we'll talk about this concept, what we have seen in the video. In the video, we have seen an ant, and that ant has some scientific name presently. But remember, this is an ant, and the common name of this ant is carpenter ant. What is the problem with this ant? The ant generally is found in the habitats where there is a dense forest with tall trees. And what do this ant do? This ant climb the canopy or the apical portion, or we can say top. The ant climbs top of the tree, and what does it do? It feeds on the leaf, and generally the tender leaf is the target of this ant. If the ant feeds on the leaf, you know very well, because we are very well known what happens when the ant starts feeding or herbivory, what happens, the effect of herbivory to the plant. Now when the ant is going to devast the plant, there is a support to the plant. The support to the plant is, just now what we have seen is a fungus. This plant is attacked or devoured by a fungus called cordyceps. It is attacked by cordyceps. So here in this, you can see the complete concept where it is showing you what is the actual height the plants are going to move from the down. And usually we can see that when the ant attacks, you know, just now we were talking about bees. When sir was talking, he said that these bees are societal animals. Similarly, the ants also, you know, you might have seen in number of situations that ants will walk in a same stream just because of pheromone or some kind of thing, you know, some chemical is released by the insect, and one insect will make the release of that compound, volatile compound, and this volatile compound will help the other insects to follow the path to reach its food. So when the ant reaches the top height, it starts devouring the leaf. Then what happens is, this ant is the ant population, whatever ant population is there, the ant population is also controlled. The ant population is also controlled by its predator. We can call it as a predator. Ant is also a predator here, and upon ant population, or to control the ant population, there is one more predator, that is the cordyceps. This cordyceps is very interesting group of fungi, belongs to a group of fungi. We'll talk all that later, but presently we'll talk about what is shown in the video, what the video is trying to tell us. The video is saying us that these ants will attack the trees. These ants will attack the trees by feeding on the leaf. Then what happens? The cordyceps. The cordyceps is the cordyceps species will have the spores from its previous life cycle, and these ant will get the spore through inhaling, and it have other mode also, as it is shown. We have the 4 stages of the complete life cycle of the predator on the ant. We are seeing the magnified image of the ant, that is carpenter ant, foraging carpenter ant, which is able to feed, and the spores are dropped by a mature fungi on the ant shell, and the spore excretes an enzyme. After falling of the spore on the ant, some enzyme is released from the spore on the shell, exterior shell of the, as you know, arthropods are very well known for their shells. Chitinous shells are very important in this organisms, you know. So when the spore, you have seen already, Muralidhar sir has told that there are receptors. Whenever specific receptor is reached, the interaction starts as if in hormone. Here also, the spores present in the air will come in contact with the exterior part of the ant, and now there is some kind of enzyme released from the spore, and it starts digesting the shell of the ant. After 2 days, the ant leaves its tree colony and climbs down to a spot where humidity and temperature are optimal for the fungus. And you might have seen carefully, if you have seen very carefully, the one ant was making the other infected ant to move from the stream. See, when once the ant get infected with the spore, the other ants identify the behavior of infected ant. That is why it is called zombie ant. Zombie ant. You know the dance. The after the infection with the spore, the ant behavior changes. The ant behavior changes. And when the ant behavior changes, the other carpenter ants which are walking in the stream will identify this one, this ant, and they will start move. I mean, they will make that infected ant to move away from the stream. See, this is a common practice happening even in a small insect. You can guess how the small insects can sense. When the ant gets infected and its behavior changes, the other ants note it and make this ant to move from the stream. They will not allow the ant to anymore stay in the stream or their whatever path is there in the path. So after 2 days, what happens when the ant is infected, or the effect of the spore is started in the ant physiology, or physiological responses begin in the ant, it will start typical movements. By sensing the typical movements, it named it as zombie ant. Now the name zombie has been made like this. So now the ant starts moving from the path and come down instead of the top part. Now it starts come down, and when it starts coming down, what happens, as it is a shady tree and a dense forest, rainforest, the entire lower area of the tree is cool and a little bit humid when compared with the top. When you see the canopy, or when you see the top part of the plant where the ant was existing, that part was actually a little bit exposed to sunlight, and it was a little bit dry. So those conditions were not suitable or favoring the growth of this spore, or it was not allowing the fungus to flourish in the host system. Now ant starts coming down, and it goes, it crawls onto the stem or underside of the leaf and bite into its mid vein, so it won't fall, then it dies. So this is happening in the particularly ant concept. The ant is infected, and then after 2 days, it will move away from its colony, and then it crawls onto the stem, or it will go underside of the leaf, and it bites the mid vein of the leaf, and it will see that it should not fall down, and then it dies. Now the story starts of the predator, that is the cordyceps. What happens to the cordyceps? Now, as the predator cordyceps was attacking the outer shell, it starts slowly moving into the internal parts of the host, that is zombie ant or carpenter ant, which is died, which is no more. So now it has shell, a part of the shell which is covering the predator, and now the spore germinates, and we can see how the long in the picture, in a picture of that ant, we can see how the ant is having from its head part, from its head part, we can see the spore germinated, and the hypha is coming out. And usually it is told that the first hypha from the spore coming out from the region which is close to the head. Now this hypha grows, and the hyphal part is called stroma, and now the mature fungus releases spores from its stroma, then spores fall to the ground and create a 10 square feet killing zone which will attack new ants. So what did you understand with this concept? The concept understood is the interaction between the prey and a predator. The prey to plant is carpenter ant. The carpenter ant attacks the many of the rainforest trees, devours the leaves, and create damage to the plant system. Upon these carpenter ants, there is a cordyceps. The cordyceps attacks the ant and control the ant population, and that is how it saves the plant from the ant attack or the ant colony. So this is a wonderful example from which we come to know about the ecosystem service. Nobody is doing nothing in this. Cordyceps has its host, that is carpenter ant. Carpenter ant is having its host, that is a rainforest tree, any tree species. And like this, one is harming the plant, and the one which is harming the plant is controlled by the microscopic form of the other component of ecosystem. Like this, the services of ecosystem, we think ant is harmful to plant, and now upon the ant there is a entomopathogen, we can say. This entomopathogen is cordyceps, Ophiocordyceps species, and that is trying to control the ant population. And the close picture of all this concept is called ecosystem service. This is called ecosystem service. So this ecosystem service is beneficial to one component of ecosystem, and maybe it is harmful for the other component of ecosystem. But this cordyceps, maybe I think most of you are, most of our teacher friends are botany background or background with fungal knowledge on fungi. So you might be knowing about cordyceps. You know very well about the cordyceps species also. As the cordyceps species Wait, let me talk about the Cordyceps species for some time. Yeah, we talk about the Cordyceps species. What I shown you, the ant one is a different species, but what I'm showing you now is about little bit systematics about the Cordyceps. There it was Ophiocordyceps and Cordyceps are the common names. It's a common genera. They are generally told. And one of the wonderful Cordyceps is the Cordyceps, which is very important, well known as Kida Jadi. I think you might have heard about this Kida Jadi earlier somewhere, or Kida Jadi is very common concept. The concept has started in the year 1997, or I don't remember exactly when it was. There was the Olympics in Sydney, and in Sydney Olympics, there was an important concept, or it is an important episode. Why it is very important episode in 1997 is because the Sydney Olympics of that particular year had several gold medals for Chinese athletes. Many of the Chinese athletes got gold medals, or in the medal list, when we see, Chinese were very top. And now, when one country gets more number of medals, it is natural that the other country will start, or the other people will want to know that how these people could get so much. I mean to say, is there any kind of drug administered to the athletes, or what happened to these athletes? Was the question. Lot of questions were there. Lot of debate was there. Though the media was not very strong at that time, like social media and all were not very active, but still the people were in a discussion that what is going on, why China has got so many, I mean to say, medals, and what was the reason. So then slowly it was come to know that these coach who are coaching the athletes have feeded the athletes with a typical kind of food. That was a mushroom. What was given as a food to the athletes, and which changed the stamina of the athletes, and that is how they couldn't get any doping positive. Without getting doping positive, the athletes' stamina was improved, and now the world has come to know that how the athletes were efficient in achieving so many medals. So from that time, though this is a common component of Chinese traditional medicine, Chinese and Tibetan, this cordyceps or caterpillar fungus is common in Chinese and Tibetan traditional medicine, but the big event achieving medals made people to more focus on this. And in many of the news channels, or one channel comes, I don't know, food or Food International, some channel comes where it was already documented how the people in Himalayan food region where people generally stay, they have a, when they come to know about this caterpillar fungus, which is very familiar in that climate, Himalayan food climate. Many of the countries, many of the our country people, Northeast people, are having a collection of these caterpillar fungus or Kida Jadi in particular season. You can check out this collection in YouTube. Videos are also there. A lot of videos are there on this. So this cordyceps, why I am trying to tell this cordyceps is, I want to correlate the one which is attacking the ants and one which is attacking here also. I want to compare. So this caterpillar fungus, that is called commonly Kida Jadi in our Indian language, And Chinese and Japanese languages also, they have their own vernacular names, and local names are also very many names are there. This belongs to phylum Ascomycota, class Ascomycetes, order Hypocereales, and family Clavicipitaceae. So we will talk about, you are seeing the picture. In this picture, you are seeing a lot of images of that particular plant. How does it look like from when it is collected from the soil? Later on, how it will be there above the ground, how it appears. These two, whatever just now we have talked, just now what we have spoke is the same one. Just now what we have seen, you have seen in the ant concept also. Whatever the ant concept is there, you have seen the ant concept. In the ant concept also, this kind of structures were found, right? This kind of structures were found in the ant concept also, and the same happens in this also. Here also it is attacking the again ant, some insect only. We'll talk about that insect now. So you see this, these are the actually pictures of Wikipedia. You can even see this Wikipedia. This is from the point of collection. This is how it is sold in market. This is how it looks in a book. And this is the picture from how the aerial part comes out after the growth after germination of spore is being shown here. So over there are, though it is belonging to Ascomycota, it is considered as a mushroom. You know well that mushrooms are usually Basidiomycota, but here also they are considering this Ascomycota member Cordyceps as a mushroom. We will not go in the debate because I'm not that detailed in this mushroom and Ascomycota concept. So what I mean to say is, over there are Cordyceps are 400 species are there. They are exotic medicinal mushroom. As I told you, these are common in Tibet and China as a traditional knowledge or traditional medicine. So Cordyceps has come from two words. Okay, one is cordy, other one is a seps. By combining these two words, Cordyceps has come. Why it is given this name is because this has come from a Latin. The origin of this is Latin origin. So it is club, the top part, and the supporting part, and the top part is head. So usually people, I told you that the many of the Northeast people will go for the collection of these cordyceps in the mountain regions of Himalayan region. So the collecting time is April to August, and usually it grows. I told you it grows in a very high altitude places, that is 3,800 meters above sea level. It should be cold. It should be grassy. It should be a meadows. All these are found in our Himalayan location. And these fungus, as I told you at the earlier form also, when I shown you the video also was saying that it is a parasite, and usually it is going to infect whatever this Cordyceps sinensis is there, or Ophiocordyceps sinensis is there. This is going to infect insect larva that is called Hepialis armoricanus. Hepialis armoricanus is a insect. Its larva is being attacked by Cordyceps, Ophiocordyceps. But in case of unilateralis, what we have seen in case of carpenter ant, the adult form of ant is attacked by the Cordyceps species, but here sinensis attacks the larval form. Okay, and then after attacking, it will make the two important structures. It is a stroma, otherwise a stipe, and the fruiting body of this is dark brown to black, and usually it has a mycelium which is yellow to brown color, and immature larva is the host, and the size on which Cordyceps grows is 6 inches below the surface of the ground. And as the fungus approaches maturity, it consumes 90% of the insect, effectively mummifying its host. In the same case, what I shown you in the case of carpenter ant. Similarly, like carpenter ant, here is a insect Hepialis species which is attacked, and it will mummify. Mummify, you know, converting mummy is what? So it mummifies the host, and 90% of the entire content of the larva is consumed. And usually when the cordyceps attack the Hepialis, the average weight of the cordyceps will be 300 to 500 milligram. And this is how is the concept which I am going to show you, the life cycle. So in this life cycle, we can see that what happens and how it happens. Actually, when we see about the cordyceps, the larval part of the Hepialis, the larval part. Actually, in the top one, you can see here is the life cycle. In this life cycle, we can see this is the adult insect, female insect of Hepialis, and it lays the egg. And now this egg, I mean to say, gives egg hatches and the larva comes out. When the larva comes out, plenty of spores are available in the soil, and now this larva is attacked by the spores, and you can see that the larva is attacked in the month of October to November, and this larva, as you know, it is called caterpillar also. So here the spores germinate, and the entire spore what germinates will make a cocoon like appearance, and now the life cycle turns. I am showing you here a picture from a book, or it is also available on internet, quite commonly available on internet. So here it is, you are seeing the routine life cycle, which is green line. The routine life cycle is a green line, what you are seeing, and the life cycle which is after attack is red. So here you are seeing the red part is the infected life cycle, and the green is the routine without infection life cycle. Two life cycles are shown to you here. And now I am talking, we are interested only in the cordyceps part of the life cycle. In October or November, so we can see the caterpillar or the larva gets attacked, and now it becomes a cocoon. And this, our by the November to April, we can see our cordyceps usually starts its germination, and then by April or May, so we can see that April to May or so, we can see that it will start making its structures. It start making its structure, and ultimately we can see the mature form of cordyceps. Whatever mature form of cordyceps is there, that is quite common or popular as the Kida Jadi is ready. And what is the use of this? So both the cordyceps, either it may be in the zombie ant or maybe in the Hepalis, it is having a wide role. It is very important in strengthening the immune system, and people say that it is also very important in reproductive functions of men especially. It will be very important in building muscles and improving physical performance, as the Chinese. The story of this cordyceps actually began there itself, building of muscles and improving of physical performance. Then it is very important for anti-age, and also reduce the restlessness, improve the respiratory functions, and also it improves the cellular oxygen intake, which is very important for athlete. And also it is important in vascular system, as it is important in regulating the blood pressure. It is strengthening the heart cardiac muscles. It is also improving the circulatory system. It promotes the functioning, normal functioning, or support the normal functioning of liver, kidneys, and it will have, I mean to say, the people who have sleeping disturbance or sleeping problem, for them it is a good, I mean to say, reported to be very good, and it generally decreases the blood pressure, and blood supply is rejuvenated, reduces the asthma and other respiratory disorders, and increases the energy and lowers the bad cholesterol, makes nutrition improve, and all so all. So today I have tried to put something in front of you, something in front of you, uh, which is maybe my target was to talk something about ecosystem service, where I have shown you the video, and hope our time is little bit near, and we'll be back again in afternoon. Please kindly join us again with us. So I want to rest my talk here itself, and hope you have able to get some new information, and uh, I want to end the meeting now, 12:15. 1:15 we'll be again back. We have to get back for our next two talks in the afternoon. So thank you. And it was I again repeat, apologize for the inconvenience what happened in the morning. It was just because of lack of knowledge of Zoom particularly. I couldn't get the correct link. I mean to say, I couldn't get back the previous link. So somehow uh, I'm thankful for one and all for sparing your time on holiday also. So by next holiday, Sunday, we will be ending our program, and almost we have done with the certificates of, when to say, certificates are made, and we are trying to get the signatures of, yesterday we have taken signature from our coordinators, director sir, and we'll be making many more lectures in the coming days. Thank you for joining with us. Thank you, and we'll get back again in the post-lunch session. Hope anybody had any query, you can ask. If not, I'll end the meeting. So hope now I need to end the meeting now. Thank you, all of you. Thank you again. Please log in with the same previous ID. You can join easily.